Welcome to our Powered by Researchers webinar series. My name is Ryan. I'm the Neuroscience Research Development Specialist here at Blackrock. So if you're new to these webinars, we're going to have a 40-minute discussion from Simon, and then afterwards we will have a 15 to 20-minute Q&A session. So you can participate in today's discussion by writing your questions into the chat box at any time. And during the Q&A, me and Simon will go through them in the order received. Awesome. So we're really excited today to be hosting Dr. Simon Jakob. He is a neurologist and principal investigator of the Translational Neurotechnology Lab at the Technical University of Munich. His lab studies complex cognitive functions in both animals and humans. And in doing so, he develops some translational techniques that are truly groundbreaking. So with that said, I'll hand it over to you, Simon.
Okay, thank you very much, Ryan. Thank you very much to all of the Blackrock team for inviting me to give this talk, and thank you very much to everybody who's attending. I'd be happy to engage in a lively discussion as well. So if you have questions, please do don't refrain from pitching them, and I'll try to get across as much information as possible. So what I want to do today is talk a little bit about what we've been doing over the past few years in terms of developing methods, as Ryan said, to sort of assess human brain function at a decent amount of detail, which means, of course, you want to home in into single neurons. And we want to do this in a way that allows us to scale these methods to ideally reach as many patients as possible, or as many hospitals and academic centers as possible. So by design, this project very early on was also about looking at the future and how this can be rolled out potentially to reach more labs than just ours, or more centers than just our university. And this was sort of a guiding principle. I'll frequently touch upon that point as I go through today's talk. So without further ado, let me start. What is absolutely dear to my heart is to make clear that this is by no means a one-man show. So all of this is a multi-year collaboration, definitely from my lab, of course, but that means that I'm not the one that is solely responsible for all of this and has done all of this type of work. So let me acknowledge first Victor Eisenkort, who's an MD PhD student, now a resident in neurosurgery, who together with me pushed this project in the very early days to a great level, where Lisa Held and Alex Utschmidt, who are doctoral students in the lab, could sort of take over some of the analysis work together with Xiaoxiong Ling, who's also a PhD student in the lab. And definitely none of this would have been possible just looking at the scientific side of life. I needed Jens Gemt, who is a neurosurgeon, moved out to his sort of own hospital now. He is the chair of the Department of Neurosurgery at the hospital, University of Hamburg now. But over the course of the last few years, while he was still with us in Munich, pushed this project massively together with me. So big thanks to all of the people that are involved. If you're interested in some of the details that we can't touch upon today, feel free to look into our preprint, which is out on bioRxiv, so has been out there for quite a few weeks now. So all of the technical details should be in there, and maybe we can touch upon one or two topics that you're interested in. All of it should be out there in writing. And if you have any questions after that, please don't refrain from just shoot me an email. It's perfectly fine. Get in touch with us. We'd love to talk about our work. So I guess at the risk of preaching to the choir, when we want to record neural activity from the human brain, we have a few options, and I stole this from a review a few years ago. Of course, you can either sort of focus on the brain from the extracranial side, which typically means you're relegated to using something non-invasive, so maybe an extracranial recording setup like an EEG system or so. With you suffer the penalties, of course, of this low spatial resolution at the added benefit, of course, of being non-invasive. Or you go to intracranial. Definitely that's the approach that you need if you want to home in into the elementary processing units of the brain, the single neurons. And you're sort of straddling between these two worlds. What I'm going to be talking about today is exclusively the intracranial world, and it's a no-brainer for that, of course, that you can't do that alone. Definitely, if you're a researcher, you need help with colleagues, specifically, of course, from the colleagues of your neurosurgical department. And this is something that, of course, is not new, not invented by us or any other lab that is close to us. This has been an idea that has been around there, floating around for a long, long time. And that even not over the last few decades, if anything. But I guess you can't just marvel at these early studies that looked at human activity at the level of single neurons. So this goes back to the mid-1950s. This is one of the earliest publications that are known to me from Seattle, University of Washington. That was Arthur Ward, who founded the Department of Neurosurgery at the university in the 1950s. And they recorded with, believe it or not, glass pipettes, like single glass pipette inserted into, in this case, this is the temporal lobe of epilepsy patients. And you can see from these early recordings that they have many of the features that we capture now in maybe a little more sophistication, but I think you have to like tip your hat to these pioneers of this type of work. So definitely we're in a field that has matured over the years, but still there is room for improvement. And that's where we, together with a few other people around the globe, hope to pitch in and sort of see how we can push things. So to sum this all up, I think if you really want to home in into the level of single neurons, you have a few options, of course. So unit activity is what you're going for. You can either use devices that are out there already for clinical purposes, and many of you will have seen this. So there are very well-established devices that are used, for example, for epilepsy patients, these depth electrodes that have been modified over the years to include, besides these macro contacts, which are clinically useful for diagnosing the focus of epilepsy, you can add these little micro wires at the tip of the electrodes in order to record single units. This has been a massively successful approach. So over the course of the last several decades now, there's been so many studies that have really promoted our understanding of human brain function at the level of single neurons. So clearly this is a good thing to go for if you're interested in human brain function at the level of neurons. But of course, like, it comes with this benefit of having a validated safety profile. But if I say this is epilepsy, well, that means that you have to probably stick either to the temporal lobe, that's where most of these recordings have been done. Now we've seen a few newer papers now, fantastic work from a few labs that are homing in into a little more special exotic sites for epilepsy. So we've seen a few medial frontal cortex recordings, which are super, super exciting. But of course, you're still bound to this idea of it has to be epilepsy and the focus of epilepsy. So if you're in a more remote brain area that isn't typically the target of these electrodes, you're a bit out of luck. So that was not the choice that we wanted to pick when we went hunting for suitable devices and electrodes. There's another option. You can, definitely if you're well versed in animal experimentation, you could just pick one of the devices that are out there. And there are so many. I just really want to cut this long story short and highlight two massive successes of the last few years, where two labs mainly have been trying, both on the East and the West Coast, sort of trying to use these very successful high channel count probes that are out there in the animal world. So these neuropixels probes that are huge successes in terms of the amount of data that you get from the electrodes, right? So in these acute recordings, they can be inserted into various parts of the brain, and providing you have the right approach and technical expertise, this looks very, very promising. And again, you just have to tip your hat to these labs that are promoting this idea and really pushing the field of using these high channel count probes for human systems. But for all of you who've read these papers, I guess it sort of seeps from the paper how difficult these experiments are. And that just, these papers are really displays of the splendid science that has been done in these labs and how collaborations can be fruitful to push the frontier. But to us, even before these probes became prominent, it was very clear that this was not the route we wanted to approach, because clearly, I think, in my opinion, these things can't be rolled out to a large community. They require such an amount of sophistication and expertise on the technical side and on the clinical side, that if you're trying to convince your local neurosurgeon to do something for you, I guess this is not the way you should go at the beginning. And this is where we stood a few years ago when we started to explore this field and see what we could use in terms of devices and how we could convince our clinical partners in our department to push this idea of getting closer to the human brain to record cell activity from ideally awake behaving patients. So we looked into a third option, and this is basically why we're here now. There is a device out there that is the UTA microelectrode array. That's been used for, again, many decades now, very successfully in various settings. So this is all human experimentation. It has been used in epilepsy patients as well, but not so much for diagnostic purposes, but more for clinical, not so much for diagnostic or clinical purposes, but really for researching into sort of the causes and the mechanisms of epileptic seizures. It's been a massive success in brain-computer interfaces. So clearly the device, even with the few years that it already has, it has this very well validated safety profile. So if you want to convince your clinician across the corridor that they should start to implement a few changes into their surgical procedures, that was our choice, right? We could very clearly argue why this would be a good device, because we can look upon many, many years of research with this type of device. That sort of convinced us it might be a good approach. What we loved as well at the beginning already was this idea of having a planar geometry, so we could be sampling X and Y dimensions, so giving us a nice sort of spatial layout, besides the option, of course, of recording of these little individual sites. We have this array with a very well-defined geometrical layout that seemed very attractive to us. And of course, I just picked two of these papers over the last many sort of huge amount of research has been out there in the BCI field. So we have motor system BCI, and now we're sort of going to the visual system as well. So very clearly, you have some flexibility here in terms of recording location. But if you look at all of these studies, and maybe that's not a big surprise to you, all these BCI studies are in the chronic setting, and then you sort of shrug and say, well, is this exclusively for the chronic setting, or why haven't people been publishing acute recordings? Have they tried but not been successful, or why is this device sort of, if anything, in this niche of being more this chronic, or in epilepsy patients, this semi-chronic device? Why haven't people been using it more actively for acute recordings? And if you talk to people, well, there is some anecdotal evidence. There's not much out there that would tell you why this might be good or not so good to use this device. But we were convinced that this might be a starting point. So what I'm going to be telling you about in the next few minutes is our experience of using the Utah microelectrode array for acute recordings. And I want to promote as well today the idea of using a special case, a special clinical case, which is awake brain surgery, but not so much now for DBS. So you might know that for Parkinson's disease or for tremor or other movement disorders, you can have burr holes placed over various parts of the brain, typically more in the anterior, the frontal lobes. And we didn't want to really do that approach because we wanted large craniotomies, so large access to the human brain, ideally in many more places than just in a few select parts of the frontal lobe, or if you look at your depth electrodes towards the temporal lobe. We wanted flexibility to go almost anywhere we wanted. So the procedure I want to promote here today, and that's how we acquired our data, was in the setting of awake brain surgery, but for tumor resection. And what awake brain surgery for tumor resection means is that patients have typically these left hemispheric tumors that sit somewhere near to these language areas of the left hemisphere, somewhere in the frontal, temporal, parietal region. Many academic centers operate these patients awake because you can electrically stimulate the surface of the brain, and if you cause some language disturbance or speech arrest, you must be very, very careful as a surgeon to resect and remove these tumor tissues. But that gives you the option of having first an awake patient, they're typically talking and conversing and communicating during the surgery, with the benefit of having a very large access sort of hole in the skull, a craniotomy, that would allow you probably to place your electrodes almost anywhere you wanted. And what we did over the course, so we sort of retrospectively went into our own sample of patients that were operated awake for brain tumors over the last few years, and we unearthed a few dozen patients and looked a little bit as to where their craniotomies were situated and where we would probably get to go with our electrodes. And pretty much, with the exception maybe of the inferior temporal lobe or the extreme occipital cortex, you can cover most of the left cortical hemisphere. Yeah, so you can see that reaches all the way from the frontal pole all the way to posterior parietal cortex, reaching into so definitely the superiormost aspects of the temporal lobe. And that's sort of the situation that you want. My lab is interested in specifically in cognitive functions. So you want to be able to cover large parts of the frontal cortex, parietal cortex, and ideally also the temporal cortex. I asked my colleagues in surgery, when they do these awake craniotomies, does the brain cool down to something like room temperature or so? Would that prevent us possibly of doing sophisticated cognitive science and recordings in our patients? They were a little unsure as to how low temperature drops. So we did a few tests, and it turns out that luckily, maybe that's what you would expect, right? The brain maintains this homeostasis. So the vasculature does this, sort of keeps the temperature stable. So you're pretty much, even though you've removed the skull, and have access direct to cortex, you're pretty much you're 37 degrees Celsius. So that ensured us, made sure that we were sort of recording in a sort of very close to physiological condition. And now what we did now, we took these Utah arrays sort of off the shelf, if you will, and we follow the very standard procedures for implantation. So there's nothing specific that we do here. You might know that these arrays come with a sort of metal pedestal where you can have access via these gold-plated pins on the pedestal surface to the individual channels of your electrode array. And these arrays have to be inserted pneumatically. So that's what you can see here. This is the array just immediately prior to insertion. Here's this pneumatic inserter. All of this is like standard procedures, equipment supplied to and introduced by Blackrock. And then you sort of trigger, so sort of you plug in the array, and then it sits comfortably in cortex. And you can already see why this might be a good thing to do, because now maybe if you go back to these other studies that use these linear probes and you have this micromanipulator system that you need to stabilize the array, you have the brain pulsating across the linear probe. And then you have all of these difficulties with movement correction. In this situation here, of course, very clearly, you have this floating condition. So the array floats on cortex, and as the brain pulsates with heartbeat, your array just pulsates up and down and sits very comfortably on top of the brain, or like gently inserted in the first few layers. So you just cover that with saline integrated strips, and there's nothing else you really have to do in that scenario. We looked a little bit as to the sort of traumatic effect of these electrodes. If you insert any form of device into a brain, you will exert mechanical damage. That's a no-brainer. So these are a few histological samples from 3 patients that we implanted just basically for testing and for histological purposes. And you can see very nicely these electrode tracts that descend all the way from the PL surface here. This is the surface of the brain all the way down. But with very few exceptions, which I'll show you in a moment, you have fantastic viable brain tissue between these sort of tracts here from the electrodes. This is a transverse section here. So you can see in sort of between these electrodes, there is no mechanical damage. And you can even sometimes see these tips, if you're very lucky and sort of cut the section, you can see how they descend all the way down to cortex. And by and large, we were very happy with the structural sort of the morphology of the brain tissue beside and sort of next to these electrode tracts. There were a few exceptions for this, as I said before. So you could, in a sort of a few of these tracts, we found some microbleeds or microhemorrhages. All of this red coloring here indicated by the arrows these are red blood cells. So you can see both along the trajectory of these or tracts of these electrodes and somewhere a little bit deeper down in cortex, you have these punctae or these little collections of red blood cells, which of course is a reflection of the fact that you are damaging vasculature capillaries as you insert these electrodes. And potentially that, of course, is one of the reasons why in the acute setting, so insert the electrode and then hope to be recording neural activity in the next 10 or 20 or 30 minutes, that might be a limiting factor, of course. And most people that you talk to, I guess, would argue that it's the mechanical damage and the irritation that you cause to the brain that prevents you from recording electrical activity in the acute setting with these microelectrodes. But by and large, structurally, we were quite happy with what we saw. And now let's go to the situation where we have now inserted the array, and I'm going to show you now a single channel recorded in this classical extracellular configuration, as you would do from your standard extracellular probe using Blackrock Neuroport system, so this clinical system for extracellular electrophysiology. This is a sort of wideband recording from a typical channel, and you can see all these wiggles. For those of you who are well versed in their electrophysiology, you know that you can decompose these extracellular signals into 2 components. And I'll show you the 2 components. So first, you can filter this and emphasize the low frequency component, which we call the local field potential. In essence, this is an oscillatory signal that sort of is a bit like the EEG measured in the intracortical configuration. That's a sort of a very standard procedure, low pass filter, and then you can do all of these nice signal processing analysis with your LFP. So you can do a time frequency decomposition, and what you can typically see is like these bouts of activity, if you will, like oscillatory bouts of activity in different frequency ranges. So all of that is like fine and exactly as you would expect. But of course, the main motivation for us in this study was to home in into the activity of individual neurons. The local field potential is a summed signal, like a population measure of many, many dendritic trees that contribute to these types of signals. So we really did want to assess spiking activity, or the ability of these electrodes to record spiking activity. And now, of course, I'm showing you one of these select channels because you can also see, besides all of these wiggles, you might have already spotted these very large deflections that look a bit like very spike shaped. And of course, if you home into the high frequency component, that's what we would then be isolating and focusing on. So this is what we want. Every single line here, so compressed in time, is an action potential of one individual neuron. And you can see they come in various sizes and shapes here. So probably we're not just recording a single cell, potentially a little bit more. This is what you want. But now we already figured very early on, if we just try the standard geometrical arrangement, as I said, most people were advocating not to use that and advising us maybe to stick with the chronic setting of these electrode arrays. So we very early on decided to implement two different experiments. And these experiments, are as follows. So we would use what we now will call the higher density, or the high, sort of, if you will, standard configuration of these arrays, which is 96 channels. So 10 by 10, 100 electrodes here with 4 of them at the corners not being active. So you have 96 channels that you can sample from. That's what you get off the shelf if you buy these electrodes from Blackrock. We were considering early on already, and that's what I'm going to show you, a systematic comparison of the standard density to something that we will now call the lower density. And what we asked Blackrock to do here is just to laser out or mill out every second row here. So in this case, that was just like an informed guess of where we wanted to start. Of course, you could have many other geometrical arrangements. We just pretty much took out every second row here, leaving us with 25 channels in this case. I'm not saying that this is like the perfect configuration, and there's still room for improvement, but this is our first guess. So what we're going to do now is compare the higher density 96 channels with the lower density 25 channels. And I'll take you through the signals that we got with these two types of electrodes. Now we'll focus first again on the low frequency component. Again, this is the local field potential. This is again a standard sort of measure of some synaptic activity, and this is an example channel. What we decided in terms of calculating a signal quality measure using the local field potential to focus on periods where there was like elevated high amplitude activity and compare that to sort of, if you will, some form of baseline wiggle down here. We formed the signal to noise ratio measure, which is given in decibels here, and we're plotting this activity over like a session in two example arrays. You can already see this is the high density array, right? This is the 96 channels here, grid-like, and then we have the grid of 25 channels. And as you can see, both arrays are quite similar. So in terms of local field potential activity expressed here in an SNR measure that I just explained, they're very similar. Yeah, so we didn't really spot too many differences between the high and low density array with regard to the ability to pick up LFP activity. And now, not even as a surprise, but sort of confirming our approach, there was a very big difference in the ability to pick up electrical activity that was isolated and coming from single neurons. So now this high frequency activity that you can see now here, this is this high frequency signal filtered, and now you can see these spikes. You can again plot a signal to noise ratio measure here. So in essence, you're taking the spike amplitude and relating that to this ongoing wiggle here, which we quantified by the root mean square. That's the baseline measurement of the fluctuation of your voltage signal. If you do that, then you can see that these two arrays, high density on the left, low density on the right, don't have the same colors. Yeah, so in pseudo color, again, we're plotting the signal to noise ratio, and you can see there's quite a big difference. With very few exceptions, we found no appreciable spiking activity on our high density arrays, but we were very happy with the yield on the lower densities. And that sort of confirms that the more channels you're trying to squash into your little patch of cortex on these arrays, the more likely is you're going to compromise your integrity of the brain in whatever measure, and that will degrade your ability to pick up high frequency spiking activity. All of this, all these electrical measures are shown with allowing the array to rest for 10 to 20 minutes. So typically insert, and then the patient is awakened for the clinical testing. And throughout that time, we just let the array sit, again, under these saline irrigated strips. So none of this, I guess, is something that if you wait much longer, you might still be able to increase your high frequency yield from the high density arrays. But this is roughly in like 10 to 20 minutes after insertion. So this is probably the time window that you have in order to start your activity, because of course, in acute brain surgery, you can't just prolong the procedure for hours just to optimize your electrical signals. So the procedure has to continue. You don't want to endanger the patient, or don't run any risks of infection or bleeding. So you have to hurry in some sense. And about 10 to 15 to 20 minutes after insertion, that's the time when you have to start. And this was quite representative of, I'll show you sort of a summary in a few slides. Next, I want to show you what happens when you hit the record button, and does that signal radically change over the course of your recording session? And what I would call a session now is 20, 30 minutes roughly. That's what a patient can do in our setting. That's also what the IRB gave us. That's, I think, a clinically decent amount of time where you're prolonging the surgery purely for research purposes. Again, we're comparing low frequencies and high frequency signals. Now, this is a comparison here from, I'm showing you two lower density arrays here, just because we have these very nice SNRs here for the high frequency component. And you can see that virtually identical SNR for the low frequencies across all channels. Yeah, so in all 25, so these are 25 lines for 25 channels here. Over the course of almost half an hour, these are pretty much straight input. So that means with this very single exception of the single spike, which I can explain later if you're interested in. This is just like an electrode pop, just a singular large amplitude artifact that we had on all of our channels and the complete array. With the exception of that, you're pretty much put, and you have this very stable SNR. So the peak-to-peak amplitudes of your field potential are very similar across your channels. Your root mean squares, so your wiggles and the sort of down states, they're very similar. And that gives you a very stable signal. And this is true as well for the high frequency component. There's a bit more variation, but again, with the exception of the single electrode pop, you get very stable signals across, in this case, this is an example of a low density array. And maybe I can show you here. So the peak to peak, that's the sort of the spiking amplitude, that stays pretty much put. We're very happy to see this, because this means that as long as you choose the right setting and get right into the brain and are happy with your signals at the beginning, it's unlikely that you're going to suffer major complications throughout the surgery. So this is something that we saw again and again, and we're sort of quite sure that providing you get it straight and right at the beginning, you could record for over the course of definitely 20 to 30 minutes. Of course, this has to do with patient stamina as well, and how the surgery goes in general, whether you hurry up or not. But electrically, as far as we can tell, there's no reason why you would have a drastic degradation or something earlier on in this session. So this is quite stable. Okay, and now what I sort of want to wrap up now for the first part is we performed a total of 13 implantations. I showed you 3 for histological purposes, really assessing the structural integrity of the brain tissue that we were recording from. Then we had a few testing surgeries where we implanted and we're trying to adjust the place where we screwed the pedestal, how we attached the cabling, how we grounded everything. So if you take like almost like 5 implantations after that, we're sort of left with 8 surgeries where we really performed the full experiment of having implantation in a patient that was later woken up and tried to record these long stretches of data. And we did half of them in the high density setting and half of them in the lower density array setting. And now, maybe that's not a big surprise, definitely to all of you who are involved in experimentation and now about the difficulties of acquiring data, and definitely in the clinical setting where you're relying as well on patients and this very complex arrangement of having a neurologist, a neurosurgeon, and anesthesiologist in the surgical room. It's quite a tricky situation. So we weren't able to acquire full data sets in all of our arrays. So due to technical reasons, where now with hindsight we know what we did wrong, but at the time this is just how the data pulls in, we weren't able to include all of our 4 arrays in the high density configuration, and we had, besides this technical glitch in one of the lower density arrays, we also had a medical complication where we sort of supposed we really had to rush everything and we weren't able to complete data acquisition. So what I'm going to show you in this slide in the next few really is sort of homing in into what we now call like the full data set of 3 high density arrays, and we have 3 lower density, but I will still exclude 1 because that was just eyeballing and we were quite sure that that third low density recording was very similar to what we had in the other two, but so to be honest here, I think we don't have the full data for that lower density recording. So I'll be showing you mainly 3 high density compared to 2 low density settings. And the good news is that they were virtually the same. So we've had very good success at just reproducing the same outcome, and we can really tell from the data quality whether we've been using a high density array or a low density array. And to cut a long story short, I already showed you this from single arrays in the previous slide. So this is a measure of signal to noise ratio in the local field potential range. And as you can see here, also from the scaling of this x-axis here, virtually the arrays are indistinguishable and the channels are always the same. So no matter which patient we had or which brain area we implanted, all of these things were very similar. Most of our implantations were frontal, and we had, most of them parietal, and we had some frontals, so these areas that I showed you before, it was very similar. And the low density arrays were indistinguishable from each other and from the high density arrays. That mimics what I showed you before in these example arrays. That again was different for the spiking activity. That is already a summary, and you know where I'm going here. So low density arrays, we had good success in capturing activity that was, you could take from single units, so spiking activity, but we were not successful in acquiring that type of signal from the high density array. So with very few exceptions, like 1 or 2 channels on one array, but we didn't really get spikes on the high density arrays. And that's something that we saw across all channels and all settings that we use. So all of this is measuring field potentials, lower frequency component versus spiking activity, high frequency component. But of course, the aim, definitely if you have good signals in the spiking range, you want to make sure that you can, from that multi-unit activity, the spikes that are contributed by many neurons, that you can isolate the activity of individual neurons. So this is what I'm going to show you on this slide here. Now we're homing into a channel. I'm showing you a single channel recorded in one of these lower density arrays, where I'm showing you that providing you know your stuff, right? If you're a well-versed electrophysiologist, you know these plots. So what you can easily do, you can separate the waveforms that you record on these single channels. And ideally, if they sort of are repetitive and same shape over the course of your 20 or 30 minutes of recording time, you can cluster them, and you can already see here you have 2 waveform shapes, or 2 clusters that appear on the single channel. Both of them are well sort of remote from the detection threshold, which is where you sort of set your level of distinguishing sort of fluctuating noise from your true spikes and action potentials. And these 2 units, as we can now call them, because they are clustered and homogeneous, they have very typical interspike interval distributions, so there is almost no violation of the refractory period here. They have very standard distributions here, and almost no spikes around these sensitive periods of 0 milliseconds, which would indicate that you're capturing multi-unit activity because you're violating the refractory period of an individual unit. So this confirms that providing you have a good signal quality, you can extend your research from just looking at multi-unit activity, so summed activity from multiple neurons, to really homing in onto the activity of single neurons. And this is, of course, not just true for one single channel that I'm picking out here, but you can reproduce that across your arrays and across your channels. So this is these lower density arrays where I'm showing you a few select channels. This is what the high frequency component looks like. And if you now go into your spike sorting, we did all of this manually here, then you can easily discern on multiple channels more than 1 unit. This is like one of the 2 ones out of the 3 that we sort of have from the lower density arrangement. Again, so if we now look into this plot, so we have on one array, we had the majority of channels had units, and on many channels, we had more than 1 or 2 units. On average, we calculated about 1 single unit per channel in the lower density configuration. And that might ring a bell to most of you. So those of you who've been using various types of extracellular electrodes, where the electrodes are spaced quite a distance, so you have independent electrodes, that's a typical yield that you would get in extracellular electrophysiology. So we're very close to what in an optimal situation, given that microelectrode array, you would be able to pick up. And I think 1 unit per channel is like a major result that we were able to obtain. And again, all of this is exclusively in the low density situation. So if you now look at the high density situation, you get, of course, that's a consequence of not being able to capture spiking activity in the first place. You're pretty much out of luck if you want to pick up your single units at least to a decent extent, using your standard 96-channel Utah microelectrode. Now let me shift gears a little bit, and this is the beauty of using these planar arrangements. As I said before, you can now look at activity, not just like in this single points of sampling, but you have this very well-defined geometrical arrangement. And now you can use these oscillatory signals that I talked to you about before, the local field potential, to say something about how neural activity travels across your little patch of cortex. So you might know that these microelectrode arrays come in the configuration of roughly 4 by 4 millimeters. So clearly that's like a worm's eye perspective on the brain, but you do have access to a little patch of cortex. And this is very interesting to see how on that patch of cortex, how activity should propagate. And this is something that you can do very nicely because you have this super clear geometrical arrangement in your Utah arrays. And what I'm showing you here is an example high density array. This now comes to your advantage that you just use field potentials for this examination. So you can use pretty much all of your awake recordings, no matter whether you have good or not so good spikes. And I'm showing you a tract here. So this is these 10 little electrodes that are spaced by 400 micrometers. They sit in this linear arrangement. And if you plot the field potential activity in these electrodes in a certain frequency band, you can see that they have this very tight temporal succession of peaks that are depicted here. And you can plot the phases of these, so the phase relationships of these simultaneously recorded LFP traces across these electrodes. And what you can then do is you can try to fit a plane to these phases. And if you have a decent fit, then you can argue that there is a very strict sort of order with which like these LFP traces reach their peak in the individual electrodes. And that's been often taken to be an indication of like a sort of a wave of activity that propagates from one side of the electrode, one side of the array to the other side of the array. So you see, sort of have a glimpse of like a wave of activity that you are measuring in oscillatory signals here that sort of sweeps across that array. You can do that very nicely, of course, in these high density arrays because you have dense spatial sampling. Yeah? And here we've plotted the amplitudes at various time points, and you can see that sort of, imagine you're sitting in like bathing in the sea or so, you can sort of really imagine how this little wave or like the rise of the sea level would sort of hit you here, and you can see how this propagates across the array from the top to the bottom. Yeah? And this array sort of comes in a certain phase gradient, so you can assess the direction with which this travels, and that is a sort of perfectly on a par with other people have seen in either chronic settings, semi-chronic with these Utah arrays and epilepsy, or even with larger spacing of the electrodes, ECOGs, for example, if you look at other settings where you're recording from the surface of the cortex. You can do that as well with the low density arrays. It's pretty much the same picture. Yeah, and I'm showing you an example here of, again, a linear arrangement. The spatial resolution is a bit lower, of course, because you have 5 channels rather than 10, but you get the idea. Yeah, so this is a great additional benefit of using these planar arrays, that besides recording ideally unit activity, you get sort of a feel of the mesoscopic environment with which these units are placed in, and that allows for, of course, a huge barrage of further analysis. There's something very nice as well. If you quantify all these types of waves, you can go into different frequency bands. All of this is sort of linking up to previous research in the semi-chronic setting with Utah arrays, or again, with ECOG in epilepsy patients. So you find there are preferred directions with which these waves propagate across the Utah arrays. Interestingly, this is frequency dependent, so it looks like a bit like in lower frequencies versus higher frequencies, these directions will change. The velocity with which these waves propagate is as well frequency dependent. And interestingly enough, the wave strength, so the fidelity with which you can fit your phase plane to the measured data, that seems to be uninfluenced by whether you're looking at lower frequencies or higher frequencies. That really means that your technique for assessing traveling waves on your arrays is robust, and you can really say a lot now about the mesoscopic system here and tell something about direction and the velocity with which these waves propagate. Both of that is true, of course, in the high and the low density situation. So this is all super nice, right? So now I've shown you that we have spiking activity, multi-unit, single unit with the appropriate array geometry. I've shown you can pick up field potentials with virtually any array that you insert in this acute brain surgery. And now what we wanted to do, my lab is interested in mechanisms of cognitive functions. So we wanted to start to relate that activity that we are measuring on our arrays to what the subject, or the patient in our case, was doing during the surgery. So we instructed our patients and trained them on a certain type of task. My background is on assessing frontal and parietal cortex function. We use typically numerical stimuli, so numbers, if you will, both in a non-symbolic setting, so where we're cueing number with the number of dots, and you can translate that in humans, of course, to symbolic number, where you give an Arabic numeral. And we've had a long-standing interest together with a few colleagues in how the primate brain represents these very abstract categories, these high-level categories, and of course the form a basis of how we perform calculations and maths in humans. And the good thing now, of course, that's the great part about doing human research. You can really go to the nitty-gritty of how we use numbers. And of course, this would be intelligible with training to a non-human primate, for example, or a non-verbal animal. But these types of symbols are exclusively human. So we can really now assess the, with our technique now, so the relationship between different notations and potentially coding strategies for any type of cognitive information. I just want to highlight numbers here, but of course the options are endless. Yeah. So the task that our subjects were doing is a working memory task. They were asked to memorize the number of dots or this Arabic numeral here over the course of a second, and then compare that memorized numerical information to a test stimulus, which came up a bit later. And as I said before, so these trials came either in the flavor of being non-symbolic, so we cued number with dots, or they could be in a symbolic notation where we cued number in Arabic numerals. And we would just really interleave these trials and hit a non-symbolic one or a symbolic one at our patients, and really try to assess the activity of single neurons, ideally, to these two notations, and see whether they have overlap in how they are coded, or whether there's no overlap. And a big question for us, who were new to this field of acute brain surgery and doing all these neural recordings, how good are patients? How good can they get? Am I promoting like a type of procedure where you might be able to get good neural signals, but your patients would just not be up to the job to perform the cognitive task you're interested in? And we were very surprised and very happy to see that even outside of the very standard neuropsychological test battery that patients typically get, or this talking that they do during the awake brain surgery that's been documented very often, these patients are really able to perform something that is quite strenuous. So if you go through these types of tasks, I really tip my hat to the patients that if you look at performance, so how good are you in this working memory task with numbers, pre versus intraoperative, there was almost no difference, a very, very slight difference. If anything, there was a small degradation by a few percent, which is exactly what you would expect. If anything, they were gently slower, but that didn't even reach significance. Only if you pooled your trials across, now this is a pool of 4 patients here. So there were very, very small differences between what they were able to do preoperative and how they performed intraoperative, which means you can really tap into these higher cognitive functions and now marry them with all of your neural signals that you have in your surgery. This is the performance that I plotted for our pool of 4 patients now over the course of the 20 to 30 minutes, as I showed you before, with the neural recordings. And now we have all of these individual patients here. And the average, you can see, it takes the patients about maybe 5 to 6 minutes or so to sort of warm up to your task. They've all been trained the day before, so they are instructed, they know what to do in this type of task. But even then, of course, the surgery is a very, very awkward situation for all people involved, in particular for the patient. So this is maybe not a big surprise that it takes a little bit to get them warmed up, but then they can keep going, right? Providing you select the right patients, they can really keep going, and you have this ability now to match the neural signals with the behavioral performance. What I'm showing you here is this sort of single subject data, and I'll cut a long story short. So you find in this field of numerical cognition, you find a very typical signature of what these patients display. So you can really tap into numerical cognition here. And the good thing is, since you're recording activity from multiple channels, and you probably won't have hundreds of patients performing your tasks, what I want to do now is just home in onto one single patient and see how much, and show you how much you can extract from a single subject with this admittedly still difficult type of data. And what we can conclude, at least showing you this is like sort of a highlight from one of these patients that had an array implanted in the inferior parietal cortex. that you can find, for example, that individual neurons in the human cortex, in the parietal cortex, have preferred numbers. So they encode, they are tuned to numerical information. What I'm showing you here is the peristimulus time histogram of one single neuron that we've isolated from a parietal cortex of one of our patients on one of the low density arrays. I'm plotting time on the x-axis here and activity of the single neuron on the y-axis. And I've color-coded the conditions where the subject was memorizing either 2, 3, 4, 6, 7, or 8 dots. And you can see there's a very ordered progression here in terms of activity. This is the phase where the epoch where the stimulus is visually presented. Now this is sort of the sensory on response of this neuron, and you can see there's a graded response. This neuron seems to be particularly interested or have heightened activity to the higher numbers here than to the lower numbers. So this is something that has been reported in non-human primates, and we've had evidence for 20 years now from the lab of Andreas Nieder, who's pushed this idea of investigating numerical cognition in the primate brain. And we now also have some very interesting temporal cortex depth electrode hippocampal and medial temporal lobe recordings that also show similar patterns of tuning. But now the good thing is about using this tumor surgery, you can really remove yourselves from these medial temporal lobe structures, as interesting as they might be, and really go to the parts of the brain where higher cognition is thought to be implemented. And now you can get closer, an inch closer towards these areas that you would be able to record from an animal setting. And now, because it's human, you can compare these non-symbolic representations with symbolic representations. Yeah? And here now I'm showing you the exact same neuron in the trials where number was cued with the Arabic numeral. And you can see this looks different. So notation matters to this neuron. Yeah? This is the non-symbolic, this is a symbolic situation, but you can still see a hint of tuning, albeit with a very notation-specific code. Yeah? So it matters whether it's this situation or this situation, but there is a hint that it might be, at least for the preferred number, similar to what you saw in the non-symbolic situation. I'll show you a different unit here. This one prefers the lower numbers, so number 1. And again, you get this very prominent, strong tuning. And if you now look at the symbolic situation here, something that you wouldn't be able to capture in an animal, again, you see a very different temporal dynamics in this, the way this neuron responds, but there is a hint that there might be at least partial overlap of the code because this unit here as well prefers the lower numbers. And what you can now do, again, this is all single subject, like 20 or 30 minutes of data, so that's not a lot. And we're sort of really trying to push how much we can get out of that data. We're training a decoder here on extracting the numerical information, so to the decoding number from the neural activity that recorded across the array. And you can readily do that in the non-symbolic situation. This is mirroring this very strong and good signals that we're able to pick up in the single neurons. And this performance is much degraded when you're trying, at least in this single subject, to decode non-symbolic number. And you can also do this cross notation. So you can train your decoder on using the non-symbolic activity and then try to see whether you can extract number using the activity for the symbolic situation. This works not particularly well, but it's a bit above chance, again, arguing for this partial overlap of code. But definitely, this is the sort of conclusion that we draw from this single subject. And this single subject, we didn't pick up an area that was completely sort of invariant to notation. So there was definitely coding for number on a single unit level, but definitely in a notation-specific manner. So not fully abstract. And this is going to be sort of my final data slide. You can then push this even further and now extend your analysis to this mesoscopic type of signal where you use the LFP and not just on single channels, but you look at the whole array, right? This dynamics across the 2D planar configuration. And I'm doing something similar. I'm showing you here how much the LFP varies or how much sort of power changes you get with changes in number. The same participant, now the same array, but now we're looking at the low frequency component. I'm extracting the explained variance, so how much of the power varies with number, how much of the power varies with notation, so was it symbolic or non-symbolic, and we're looking at the interaction as well. And you can see that mirrors a little bit what you saw in the spiking activity on the previous slide, that after the presentation of these numbers, you get, in this case, in the sort of higher frequencies, the gamma band, you get quite an amount of variation of power with number. You get a fair amount of variation with notation, so symbolic or non-symbolic, and you get a fair amount, of course, of interaction. But now the interesting part now comes here is if you plot the activity, in that case this is power as a function of number for these two conditions, you will see that I'm plotting gamma and beta activity here. So this would be sort of the range here, just 100 and below, and then the beta band would be somewhere around the 20 or 30 range here. You can see that very similar to what we saw in the unit activity, we see this increase of power in the symbolic and not in the non-symbolic, and not so much in the symbolic situation. So very similar to the spiking activity, this is a brain area, of course, also known as P, maybe not a big surprise, that would care a lot about coding of number, but more now, and this is interesting, not in this peaked tuning fashion that we had previously, but this seems to be a very general effect of just monotonic increase of power, and specifically in the gamma band, a little bit more in the beta as well, but just scaled with number. The higher the number, the higher your power. And I'm plotting the same channels that we looked at in terms of single unit activity, and you see a different pattern, right? Remember that channel that had that tuning to number 1? So now all of our channels that we've looked at in this single participant had tuning to higher number. So it's not like the gamma activity would follow the exact same coding regime as the single units. And maybe that's also not a big surprise because, of course, oscillatory signals are summed neural activity. But this is nice for us because now we are opening up this idea of being able, of course, to compare the lower frequencies and the higher frequencies components, and they clearly index different aspects of numerical processing. If they do that for number, well, I think it's a no-brainer to believe that this might be true as well for many other different cognitive functions. And now, again, exploiting this 2D arrangement and this wave idea. So what you can now, of course, also plot is you can plot how many waves you have, how strong they are, if there's any differences. So we saw strong waves in all of these different types of trials. So there was no particular difference between wave strength, between symbolic or non-symbolic, or between higher and lower numbers. But there was a very interesting finding here that in the non-symbolic situation, we have this faster propagation of the waves as the numbers were higher. This is an effect that didn't reach significance in the symbolic condition. So you're exploiting a different aspect of your brain signal here, and something that goes beyond looking at the little pinpointed signal that you have from a single channel with spiking activity. I want to stop here. Just again, I gave you a glimpse with what the cognition part can be. Of course, I focused more on the electrophysiology, on the neuroscience part, and just leave you with this take home that my team sort of have been working on our conclusions from this type of project that we've been conducting over the last few years. So I think if you are interested in assessing the neural correlates of human cognitive functions, I think it's well worth, if you have access to a surgical department, it's really worth teaming up and pitching this idea of going to awake brain surgery, and not so much for movement disorders, but for tumor resection. This has been something that is not at the forefront previously of cognitive neuroscience, of course, because of the difficulties of accessing these patients and providing these collaborations. But we are really, really happy with sort of seeing how this panned out over the years, because these large craniotomies do provide direct access and broad access to the left cortical hemisphere. And that really removes you from the sort of having to stick, for example, to the temporal lobe or the medial frontal cortex. And ideally, you can access many other parts of the cognitive system. I was promoting this idea in the large majority of my talk, that your off-the-shelf electrodes that you can get in these microelectrode configurations are suitable in general for intracortical recordings in the acute setting. So we didn't encounter any single adverse event that was related to the implantation of the electrode. So that single medical complication that I talked about before, that wasn't anything at all to do with what we were doing together with the patient. So we would definitely think over the course of our 13 surgeries or so that all of this is safe and it's practicable. So we were able to convince and educate and train together with our surgeons to perform these types of procedures. And we now have this team set up that is quite well versed in these intracortical acute microelectrode array recordings. I also pitched the idea that you have a choice, of course, in general, of which electrode you want to pick for your type of research. There might be lots of pros and cons for individual situations. But for us, this was as an eye-opener that you can use these microelectrode arrays and exploit the planar geometry, but you have to be aware that if you use the sort of off-the-shelf standard configuration, you are compromising signal quality here. most likely because of the traumatic injury and the sort of pressure you exert when you implant these arrays in the very dense situation. You get much more dimpling than when you do this in the lower density configuration. You have to sort of compromise a little bit. And if you want to go for the standard 96 configuration, I think you're going to suffer a little bit on the spiking activity. If you're interested in field potentials and these waves and the propagation, they're perfectly fine. But for us, it's been a big wish to really go to the unit activity. And first, it's been a game changer to use these lower density arrays rather than the high density arrays. And the big motivation for us at the start, and this is how I'm going to finish my talk now, is that my hope is that some of these technologies will scale. So of course, you always need people that promote these ideas and that push the frontier. And I talked about these amazing contributions from the NeuroPixels recordings. But ultimately, you have to make sure that your idea spills over, right? You want people to be on board. You want to increase the critical mass of getting people on board and doing similar type of research. And for us, it's been easy to do this, providing, of course, you get the right people, because we were using a device that is established. It has a validated safety profile. Our research isn't changing any, we have no additional data to report adverse events on these types of recordings. But we think that now, because the technique is almost off the shelf, with a few tweaks, it's quite easy to get your surgical colleagues to be interested in this type of research. And you don't need too many changes. I'm not saying this is a turnkey situation, right? So far from that, I think anybody who's ever been in an OR and recorded from an awake behaving human from cortex knows these types of things. So I want to be honest, of course, right? It's not easy research, but because the device is simpler and the technology that is behind it is more robust, the hope would be this could be scaled up. And because you have this broad access to the human cortex, you might be able to tap into mechanisms of many other different cognitive functions. And I just highlighted the idea of the numerical, the number sense in my talk. So I want to go full circle again and close today's talk with thanking the people that did the work together with me. So this is nothing that I did alone. I highlighted Victor, Hyung, Alex, and Lisa, who contributed from my lab. I want to thank everybody else for helpful discussions and for being supportive over the years regarding this type of research. This wouldn't have been possible at all without my friends and colleagues from the Department of Neurosurgery, Bernhard Meyer, the chair of the department, Jens Gemt, my colleague and friend who performed these surgeries together with me. They were absolutely instrumental. And if anything, I think you need friends and colleagues to pitch in with you. But if you do that, and if you find the right people, I think great things can arise. So thank you very much for listening. I'd be happy to take the questions.
All right, wonderful. Thank you, Simon. That was a really well done and really thorough presentation, so we really appreciate it. Just for the sake of time, I want to just quickly mention that if you need to go, this webinar is being recorded, so we can provide the recording to you if you need. And before you leave, we would really appreciate if you could take a few minutes just to fill out our survey that you will get at the end of this. With that, we have a ton of questions, so I'll go ahead and just jump in here. The first question comes from Stephen, and he asks, was the array target location determined completely through pre-op imaging and testing?
No, it was largely, yes, of course. So you know where you placed your craniotomy, and we didn't change any of these medical necessities. But of course, the intraoperative site and the situation, the scenario in the OR determines as well where to place the array. So you can have sophisticated MR imaging and other techniques, functional localizing if you want to, but ultimately it's the decision of the surgeon, and it's up to the surgeon to decide where to put the array. There might be like vasculature or a vein or some other pathology where you wouldn't want to insert the array. So we always took both the preoperative assessment, imaging, mainly structural, and the intraoperative situation into account, and then sort of used both to decide where to put the array. Of course, we didn't want to plonk it onto the tumor itself, so we always made sure it was in like a safety margin around the tumor, the margin that would be resected together with the tumor. But it was always you will only really know during the surgery where you will end up.
All right, the next question is from Abhilash. He asks, how do you detect the high and low activity states, HMM, and did you try this during sleep or anesthesia?
So we have some recordings during sleep and anesthesia, but everything that I showed you was in the awake situation. So it's not like the sleep activity that I showed you. If I may, I'll just go back. I guess your question was related to the field potential, high and low states. So I'm not saying this is like the up and down of maybe an anesthetized preparation. It's really just using the amplitude, so the envelope here of the local field potential, and just putting a mean across the envelope. And if it's above the mean, we call it a high activity state, providing it survives for a long enough time. And everything below we call the low activity state. This is a bit of a, it's a workaround. So when we were shopping for ideas of quantifying signal strength in the local field potential, there wasn't much that we thought was particularly useful. So we were inspired by a few other people, but I'm really saying a few, like 2 or 3 papers that used similar approaches. I guess you could come up with many different options here, but again, I'm not promoting this as the up and down state of something that was similar to sleep or deep anesthesia. So.
The next question is from Hatra from Blackrock. She says, thank you for your presentations, very inspiring work. And she asks, how long did you usually wait before you started the recordings after implantation? And did you notice that the signal quality increased when there was more time between the implant moment and the start of the recording?
So second question, definitely yes. I guess the longer you can wait, the better it is. Of course, this is the problem in acute brain surgery. You can't wait for as long as you want to. So if we had waited for maybe an hour or two, I guess it would have been better and better and better. And we definitely saw over the course of a few minutes, like these spikes slowly emerge. This is something that people have been recording in these linear probes that have much smaller footprints for the single shank. So they've not really seen that, and that makes a lot of sense, right? The smaller your array, the smaller your configuration is, the faster you will pick up units. And that's a big advantage of these NeuroPixels probes, for example. You can insert them and you get units quite quickly. So we typically had to wait 10 to 20 minutes. The good thing is we didn't really prolong the surgery, because the way we designed the experiment is we inserted just before we woke up the patient, and we used the time where the patient woke up before they are really up to performing the cognitive task to just let the array settle. So we weren't really compromising on the duration of the surgery, but definitely, yes, it makes a difference as to when you hit the recording button, and you want to wait as long as possible, but there will be limits to that.
Great. As long as you're okay on time, there's plenty more questions.
Yes,
you
definitely. Keep
great.
coming. I'm fine.
Awesome. The next question is from Chadwick, and he asks, were you able to measure impedances, and were they predictive of signal quality? And then he sort of expands on it by asking, if you were unable to measure impedances, can you please point out any technical or practical limitations?
So we always aimed for measuring impedances. That was always one of the very first steps after implantation. So what we found was, we found that they weren't very predictive of how the surgery would then continue. So typically, and I think many people have seen that, have inserted these types of metal electrodes into a biological preparation, namely a brain. They've seen impedances being very, very high at the beginning. And then over the course of, typically if it's a semi-chronic setting, over the course of several days or so, the impedances come down. So what we figured over the course of doing this for 13 different implantations, that although we measured impedances every single time, I can't really say that there was a strict correlation as to they were very predictive of how the surgery would continue. Very typically, we had higher impedances than what are normally supposed to be like the standard for these types of arrays. So most of these were iridium oxide, so we're somewhere definitely below the 100 kilo ohm range. We typically didn't see that. If you get them low at the beginning, that's like a good sign. That means, I guess, you've hit the brain and everything else is fine. But it depends on so much. These impedances depend on not just the arrays being in brain and you not hitting a capillary or like a little air bubble, whatever there can be. It also depends on, of course, closing the circuit. So your pedestal, which we screwed onto the skull, has to be not just in mechanical contact with the skull, but it also has to be an electrically conductive interface. So that's something that we noticed a lot, that impedances would vary a lot with how we irrigated, for example, the base of the pedestal. And if you wrap like little irrigated little drapes or so around the pedestal, you could lower impedances, which of course has nothing to do with the situation the electrodes are in brain. Yeah. And given the complexity of impedances, to cut a long story short, I think we would always continue to measure them, but I wouldn't base my decision on whether to record or not purely on the impedances. You just look at your spiking activity or your field potentials. And if you're happy and if you're well-versed electrophysiologist, you'll quickly see what is a good or bad signal, and you'll just hit record depending on what you see on the spike panel or on the LFP panel.
Okay, yeah, that makes sense. Thank you. The next question is from Elise. She says, "Thank you for the great talk. And the high-density arrays show worse signal-to-noise ratio than the low-density arrays in high frequencies. So could you expand on why you think this is?"
Ultimately, I don't have proof for any of my notions here, but if you insert an array into brain and you compare the high and the low density just really slides in, and the high density needs a fair amount of push. Of course, all of this is pneumatic, right? But you can easily, I think the argument is mechanical, that the amount of tension and dimpling and everything that is created, I'm not an engineer or biomedical engineer, I guess you would have much better terms for these types of things, but ultimately I think it's purely the mechanical irritation that you exert in brain when you're trying to insert these high density arrays. So it's not so much the channel count. I guess it's really about density. It's about electrodes per surface area. If you were to space your 96 channels over maybe 12 millimeters and not 4 millimeters, I think you would get 96 channels of very, very decent activity. And that's ultimately the big advantage, I think, of having these linear probes, which are very thin, like a few hundred micrometers or so at most, where you're really just piercing the pia and the brain in a single tract. And that's the reason why you get this fantastic signal-to-noise ratio very early on, without all of this mechanical irritation subsiding, with, of course, all of these caveats and downsides of having the brain pulsate across the array and the movement artifacts. But I would argue it's a mechanical irritation. It's a local effect. And that's my take why the lower is better than the high density.
Great, thank you. So we have many more questions.
I'll just add, it would be great to see this structurally as well. So, of course, if you had way more recordings than we, and if you had histology and perfect sample staining and preparation every time, ultimately your hope, of course, would be you would see some structural correlate of the higher density arrays exerting more damage and more like breakage of capillaries or like dissolution of brain tissue or something than the low density arrays. But unfortunately, structurally, we just made sure that we were safe and we didn't have these huge bleeds, but we don't have enough structural data to verify that. So it's still a hypothesis.
Okay, got it. So yeah, that's great. So we have quite a few more questions. I think just for the sake of time, I'll go ahead and collect those questions and send them to you in an email, and then I'll give you the information so you can reach out individually. But just to end here, I did have a question myself. At the end of your talk, you talked about the like the scalability of these procedures and this application. I'm just curious, with your own lab, what are your next steps with this project, and where do you see this going in the future?
So we have now we've been shedding all of the problems. We've gone through wading through all of these problems, and now we think we know what the knobs are that we have to turn. So now we're in the situation of ideally being able to include every patient that is operated awake for brain tumor resection for this type of research, providing, of course, the patient is suitable and consents in all of these givens, right? So, but then that would mean, like, for example, for our department, that would mean that you can easily get to like a dozen patients or so a year. That's already what I call scalable in a sense. But, and if you look at other clinical centers that do these surgeries, this is not like a exceedingly rare procedure, right? It's a bit of a matter of how clinical departments decide on where to put their focus. But I think because you are now homing in onto this group of tumor patients, you have access to a much larger group of patients than, of course, if you were to use the arrays for their original purpose, which maybe is the brain-computer interface. Also, if you take those spinal cord lesion patients, how many spinal cord lesion patients have been implanted with Utah arrays over the course of the last 10 to 20 years? It's, I don't know, you probably know the numbers better than me, but it's somewhere on the order of a few dozen at the most. Now, if you take your epilepsy patients and the semi-chronic recordings, then you have more patients, but still it's quite a low amount. Right? And the good thing is about the tumor patients, that's a group where you could capitalize on. Providing they consent, you can include them and do research on many different parts of cortical hemisphere function. The added benefit, actually, of using the Utah array, and that was, I didn't say this explicitly, but this was a design consideration for myself. If I train a surgeon or a department, my colleagues in neurosurgery, to use these arrays, there was the benefit, of course, going chronic in a second step. So now you have the advantage of being able to use the arrays in the acute situation. That's our contribution. But of course, we know they perform exceedingly well and are the gold standard for all of these chronic BCI or other types of applications. And now that we know the technology, we know the arrays, the surgeon knows how to use these arrays, we can now open up a second discipline, which is now chronic implantation, and which we're actually already doing now. We have been implanting these arrays chronically as well. And this also stems from the fact that we've been using them in the surgery. So we've been using acute recordings, besides for the purpose of this study, also to train our surgeons and getting familiar with the arrays. That's an aspect I cherish a lot. And if you, of course, look to the other types of arrays, they are relegated to certain brain areas or maybe to certain settings where you can't just cement in the array and use it for different purposes. So this UTA array for us is like a multi-purpose workhorse now, where you can go into various different fields, and we are now pushing both on the acute and on the chronic side.
Got it. Great. Yeah, well, thank you so much, Simon. We really appreciate your time. For the rest of you who did ask questions, like I said, we'll collect those and we'll get them out to you via email. So thank you, everybody, for attending. And yeah, thanks again, Simon. We appreciate it.
Okay. Thanks, Ryan. Thanks to everybody else. Thanks for attending. It was a pleasure.